So welcome everybody. If you'd like to put your name in the chat, let us know where you're from. We'd like to see that. This is going to be recorded. Michael will send out the slides and the recording link in a bit after the call, like we usually do. We're very excited today that our dear Richard from TAG, our Encyclopedia Britannica, is going to give us a cure overview. And it's been working very hard on it. So I think it's going to be very interesting. I think I said a second ago we thought this might be our last webinar, but
Hmm.
we're hoping that we will continue to have sort of bridge funding for the rest for a year, and we're going to keep at it as long as we can. And hopefully you'll continue to join us. I'd like to say that you should put your questions in the chat. Michael will monitor that after Richard's presentation. We can have a nice long question and answer session. And about three-quarters of the way through, Michael will put up a screen where you can, vote on what you thought of the session. It's a quick interactive thing. Next month, we're going to have Dr. Steve Deeks, who'll be talking about his gene therapy CAR T cell trial. That's going to be on July the 14th, same time, same Tuesday kind of thing. And we're very excited about that as well. So without further ado, let's let Richard get started because he's got a lot to talk about and he's worked very hard for us. So take it away, Richard.
Thanks so much, Linda. And hopefully I didn't overpromise too much in the description for what I'm going to be able to cover today. I'll try and get through a sort of substantial, or at least some of it. The main focus really will be on treatment action groups listing of ongoing HIV cure-related research. But I'll start with some of the examples that sort of have spurred the HIV cure research effort, then switch to the listing and the types of interventions that are being studied right now, where those studies are happening, and what we've been able to sort of capture in terms of trying to track participant demographics, and then talk a little bit about the areas that seem to be showing a lot of promise at the moment, and briefly summarize challenges. So I think everybody is probably aware that there have been some cases of HIV cures achieved by stem cell transplantation for a serious cancer diagnosis. Very honored to have Mark Frank on the call today. The first was Timothy Ray Brown, who's sadly no longer with us. He first reported in 2008 at the Conference on Retroviruses and Opportunistic Infections in a poster that actually nobody noticed, apart from Martin Delaney from Project Inform, who spotted it and wrote about it and was very excited about it. And the most recent was a Canadian case that was reported earlier this year for 11 in total. And it's great to get to a point where it maybe started to be possible to lose count. There are two women now as well and the remainder are men. There's a great summary on the AMFAR website called Curing HIV, How Far Have We Come? And I'll share these slides with Michael so he can include them when he posts the video in the notes under the video, and then you'll have access to the links that are included.
Great.
And I think this has been discussed quite a lot on these calls over time, but obviously this is not really a practical approach for most people. Most of these individuals received stem cells from donors that had genetic resistance to most HIV strains. Others didn't have that donor mutation, and it seems that the depletion of their existing immune system by both the sort of chemotherapy drugs that are used to deplete to make space for the transplant, and also graft versus host disease that can happen when the new immune system starts clearing out any remainder of the old immune system. That seems to have been key in clearing the HIV infected cells in these individuals, leading to a long-term cure. Like I said, it's unfortunately not a practical approach. And people forget sometimes that the post-transplant care can be burdensome as well, because it can involve immune suppressants and things like that aren't necessarily easy to deal with. And then to move on to the other encouraging cases, elite controllers are people that have been known about for some time. It's a rare group of people with HIV, I think estimated at less than 1%, who naturally can control virus to undetectable levels without needing treatment. It's associated with particular immune system genes called HLA genes and strong, what they call CD8 killer T cells that are capable of killing virus-infected cells. And in more recently, although elite controllers have been known about for some time, there have been 2 reports of individuals where they appear to have cleared all the intact and viable HIV in their bodies. So really a natural cure has been achieved. And this kind of analysis has been made possible by advances in the tools for analyzing the virus that persists in people's bodies. And it allows researchers to distinguish viruses that are intact and capable of replicating and those that are defective and sort of damaged in various ways. And most of the time, the majority of viruses kind of get trampled by the immune system and have defects. And it's really the intact viruses that are most important and that seem to have been gone from these 2 cases. The first was Lorene Willenberg, who sadly died earlier this year from lung cancer. And when Lorene received the cancer treatment and the immune suppression, and no HIV came back, which was really viewed as confirmation that HIV had been fully cleared from her body. Lorene was an amazing advocate, set up a foundation for elite controllers, and I've included links here to a fairly recent 7 months ago previous webinar in this series that talked to Lorene and researchers that have worked with Dr. Zhu Yu. And Lorene was also lead author on an advocacy paper about the situation we currently face with HIV research that was published in Journal of Infectious Diseases. The second case is an Argentinian woman from a location called Esperanza, which means hope. And similarly, she was an elite controller who now appears to have cleared all the viable HIV from her body, just establishing the principle that this is possible, which is obviously very encouraging. And it sometimes maybe gets a little bit forgotten that post-treatment controllers have been reported for quite a long while. Initial reports appeared in the late 1990s of people typically had started antiretroviral therapy early, and were experiencing periods of viral control after the antiretroviral therapy was interrupted. And Walter Armstrong, the editor of POS magazine at the time, asked me to write a piece, I think this is from 1998, called Mission Remission, that was his title. And obviously that term has come back around again a bit. And actually it was this article that Lorene saw when I first met Lorene. It's because she emailed after the article appeared. And these cases, at least established that this might be possible, but there's going to need to be additional interventions to make the suppression better and extend the time. And more recently, there's been more reports of this happening in both adults and infants. The Visconti cohort of early treated in people in France has around 14 to 20 people, I think, that have controlled viral load after an antiretroviral therapy interruption for a number of years. The AIDS Clinical Trials Group, as it used to be known, or Advancing Clinical Therapeutics globally, as it's now known, has analyzed their studies, found 67 cases of post-treatment control, and 20% of those 67 people were actually controlled to under 400 for more than 5 years. So again, sort of encouraging. There is some evidence for both elite controllers and post-treatment controllers that it may be a more frequent phenomenon in women, people who were female at birth. In infants, there's a small study that suggests it could be the opposite, that actually male infants may be more likely to experience this phenomenon, but I think the data is still very limited at the moment. Again, these results are summarized really well on the AMFAR resource page. And then to switch gears into sort of what's happening in clinical trials right now, based on the encouraging signs that that a cure might be possible. A treatment action group where I've been fortunate to work for quite a long while. We've been trying to monitor the research since 2014. Clinical research just means research in a clinic that involves people as opposed to laboratory or animal research. We mainly draw the information from clinical trial registries, primarily the ones supported by I think it's National Institutes of Health, the National Library of Medicine, clinicaltrials.gov. Also, there are also in various international registries that we check intermittently. And this just shows the first page of what is now a 57-page document, because we keep track of the trials after they're completed. And this is an example of the kind of information, we try and track results and add links to results as they're presented. The shading in the two entries at the bottom are indicative of a study, including an analytical treatment interruption. I took a look at the number of newly registered studies by year. I don't think these necessarily represent trends lines, particularly. It's kind of averaging around 15 to 20 newly registered studies per year. There was a bit of a dip there. I think that's COVID related. And there is some fluctuation. There hasn't been many observational studies. Those are studies where people take samples and measure various things, but don't give people interventions. And those seem to have dropped off a little bit. But also, I don't think they necessarily are required to register those studies. So we may miss some observational studies. And then to get to where we are right now in the listing, there are 79 trials, 76 adult, 3 pediatric. Most, as you would imagine, are early phase. Phases are used to generally sort of divide up how far along an intervention that's being studied is. But in this case, the later phases really relate to the populations being studied, and unfortunately don't mean that there's a cure intervention being tested for efficacy. Most of this is still quite early work. There's 42 observational studies, just 1 pediatric there, and 37 studies involve analytical treatment interruptions, which I think it may be about the highest that there's been. It's about 30% of all the studies that are asking people, not mandatory, but asking people to interrupt therapy to analyze what happens to the viral load after treatment is stopped. And this one is a little bit of an eye test in terms of all the various names of the different types of interventions that are being studied. What I think is important to draw attention to here is that, not too surprisingly, broadly neutralizing antibodies that you may have heard quite a lot about already are one of the most frequently studied interventions at the moment. Combinations similar to what happened with antiretroviral therapy. There's a lot of thought that a combination is likely to be needed to achieve a long-term suppression of viral load. And gene therapies are next in terms of the most frequent types of studies, although that's a little bit skewed because the Food and Drug Administration requires that people do very long-term follow-up of people that have received gene therapies. So I think there's 3 studies there that I'm not sure the companies even still exist anymore that sponsored the original studies, but it's required that follow-up goes on till into the 2030s to make sure that there's no undue safety risks that emerge. And on this slide, what I've tried to do is just highlight the categories of interventions where the studies are using a treatment interruption, because I think that gives a little hint that the researchers are hoping that they will see something useful. It's very common with broadly neutralizing antibody studies and with combinations where almost all the current studies are using treatment interruptions to assess if the interventions have modified the person's ability to control viral load. And there's a variety of others that do plan on using a treatment interruption. Immune checkpoint inhibitors that have shown success in cancers were more extent. There used to be more of studies of those. And the drug company AbbVie did a very large study, but unfortunately weren't really able to show long-term control, and there were immune-mediated adverse events that probably are going to preclude using that approach in its current form. The one study that remains ongoing is using a low-dose PD-1 inhibitor. I should note also that the studies for people, the gene therapies for people with HIV and cancers, that the treatment interruptions are only done in those studies under very specific circumstances if specific parameters are met regarding the uptake of the gene therapy. And I'm not sure if treatment interruptions have happened in those studies. Just a quick look at the primary sponsors of the research. This is kind of difficult based just on looking at the registry entries to assess where the money, the funding support is coming from. I grouped together all the kind of networks and different institutes that are supported by the National Institutes of Health in the US, which includes Impact of the Pediatric Network, BTN, the Vaccine Network, and various other NIH institutes that are sponsoring HIV cure research studies. That's a fairly large slice of the pie. The biggest is academic medical centers, but again, NIH funding is likely to be involved in many of those studies. USMHRP is the US military HIV research program. ANRS is the French network for HIV research. CAPRISA is in South Africa. IAVI is a vaccine organization, and industry is sponsoring a fairly small amount of studies at the moment, 12. But I think the sort of message here is that NIH funding is obviously really important for this research. And just to drill down a little bit into which companies are sponsoring HIV cure research, I've highlighted probably the three biggest ones. BioNTech was a biotechnology company. I think it's probably grown in size because of its successful COVID vaccine, and it's sponsoring a single study of a new broadly neutralizing antibody. Merck is sponsoring a trial of something called a targeted activator of cell kill, or TACK molecule, which I'll talk a bit more about in a little while. And VIV Healthcare is sponsoring two studies, one of a broadly neutralizing antibody and one of a broadly neutralizing antibody combined with its entry inhibitor drug fostemsavir. And those are really the biggest companies sponsoring research right now. Gilead has been a big supporter of cure research and has conducted many studies. They don't have anything ongoing right now. I don't know whether, hopefully not too much to read into that, but maybe something to keep an eye on. And study site locations. So this was my attempt to use Excel to create a pie chart, which is not necessarily a strong point, as you can probably see. I think the not too surprising thing here is that still the United States is the major location for studies, and the distribution around the rest of the world is more limited. Another way of looking at this is sort of breaking into regions, and again, sort of United States, North America. I think there's one Canada and one Puerto Rico in there, and Europe are the major sites, with Asia Pacific, African continent, and South America making up a smaller portion. Obviously, some studies have multiple country locations, particularly like the Impact early treatment study in infants. So there's 158 different sites for the 121 current ongoing studies. One of the things that the listing allows us to do is, as results are presented or published for studies in the listing, we extract if information is reported, which isn't always the case, on participant demographics. We can extract that information and sort of track it over time. We started doing this in 2018, and we've been doing it ever since. We now have a total of 8,471 participants that have participated in HIV cure research in this time. as you can see from this table, it's still very skewed with women are underrepresented compared to the global epidemic, seriously underrepresented at 18.9%. Transgender people very much underrepresented, and the kind of horrific bigotry we're facing, and this sort of anti-science insistence somehow that transgender people don't exist, that's happening not just in the United States but elsewhere, is can only further damage the potential for involving transgender people in research, which is problematic on multiple levels, because when there are issues around sex differences with interventions, one population that can really help understand what's going on is people receiving gender affirming care, because it helps understand both the immunological effects of that care in the transgender person, but also what the contributors might be to the sex differences that are described, because you need to try and better understand whether it's coming from chromosomal, XY, or whether it's hormonal. And transgender people could have a big role to play if they weren't facing such horrendous oppression right now. Similarly, race and ethnic diversity is not optimal either. I won't read through all the slides here again. I will share these afterwards. And I should have actually included the number of studies that this is, but I think it's around maybe 120 or so, but I'm not totally sure. And then sort of shifting gears again from the tag listing into what out of all those different types of interventions may be showing some promise. I think broadly neutralizing antibodies we've already heard about quite a bit about. There's been evidence now that these are, to back up a little bit, these are antibodies that have been identified in people with HIV that have unusually strong activity against a broad number of global HIV variants. And they may also have the capacity to enhance immune responses to HIV, as well as directly suppressing viral replication. There have been multiple studies. Rockefeller has been a leader. That's where a number of these antibodies were discovered. University of Aarhus in Denmark, Wistar in Philadelphia. And there's been some evidence of individual cases of extended post-treatment control, accelerated clearance of the persistent HIV reservoir. But there are some issues around because BNAbs target the outer part of HIV, which is very variable. Baseline resistance can happen in people that have never been exposed to a BNAb. And so pre-trial sensitivity screening is often needed to sort of select participants, which is not ideal. Perhaps one thing for the future that I think there's some hope about is the potential, rather than infusing or subcutaneously injecting broadly neutralizing antibodies, using gene therapy vectors, particularly something called adeno-associated virus. There's been one study at the National Institutes of Health looking at that's published some results, and they were able to generate the antibody in a person's body after an injection, but only at a very low level. And obviously, if it was possible to do a one shot that could deliver a high level of antibody in a sustained way, that would be a much better potential way of delivering antibodies. And there is this link goes to it's a preprint, so it hasn't been peer reviewed, but it's a sort of depot implant technology that they think would be able to sustain delivery of BNAbs at high levels. And it might also be useful because in addition to BNAbs, there are some other antibodies in development that don't target HIV directly, but instead target CCR5 as a way of blocking the virus. So there may be a variety of antibodies that could be delivered in this way to suppress HIV. The largest trial of BNAb so far that's been presented is the Rio trial, primarily in the UK. I think it's adding some additional sites in Europe now. The results were just published recently in the Lancet HIV. The initial round of results is 68 all cisgender men, and there was the initial comparison is between people that received two BNAbs versus those that received placebo, and there was a really big difference in the ability to control viral load after a treatment interruption. With 13 people controlling for 48 weeks and 7 out of 34 that received the BNAbs controlling for 72 weeks, compared to just two in the placebo group. Although those two people did control for 72 weeks, which I think, there's some uncertainty around the ethics of including placebos. But this result sort of shows that you can have these cases of people that control without any additional intervention. And so this comparison probably was necessary. Simon Collins, the UK community advocate, was very involved in the design of this study. And one of its novel aspects is that the placebo group gets switched and has an opportunity to receive BNAbs in the second phase of the study. So that's how they sort of address that concern about withholding the intervention. There's been some presentations at CROI and in preprints reporting that enhanced T cell responses seem to have contributed to this control that was seen, and also that people's natural antibodies were contributing, what they call autologous, which just means a person's own antibodies against HIV were suppressing. And also the decay, the decline in the size of the HIV reservoir was faster in these study participants that received BNAbs versus historical sort of data on that. And then the other area where I think some promising results have been seen is with combination approaches. The FRESH cohort is based in South Africa. So, it's research funded by the Reagan Institute at Mass General. Involves a cohort of young women who started antiretroviral therapy very soon after HIV acquisition. And Gilead sponsored a trial that recruited 20 women and administered two BNAbs and what they call a toll-like receptor 7 agonist. And a toll-like receptor is a kind of signaling receptor that's on the outside of some immune cells. And when an agonist is something that binds to it and sort of boosts the activity, the idea is it will boost the antiviral activity of the cell, I think. And they did see delayed viral rebound in a fairly substantial proportion of the participants. And 4 had extended post-treatment control. And I believe most are still being monitored, but I haven't. The last update I saw, I think, was about a year ago. And again, there's a great report by Simon Collins for iBase that goes into a lot of the detail about the trial results. Another study that has received a fair bit of attention recently was conducted at the University of California, San Francisco, a small immunotherapy trial that was open label. Everybody received the interventions, which were quite complex and I think quite sort of difficult for both the participants and the study nurses to get everybody on schedule. I think COVID was happening around this time as well. So it was a real sort of logistical triumph on the part of everybody to get the study to happen and to work. And there was an unusually high degree of post-treatment control in this small population. And there's one individual who has suppressed viral load for over a year. And I think then restarted and stopped again. And the webinar that I mentioned earlier that involves Lorraine Zhu, the doctor, also talks about some of their preliminary studies looking at the reservoir in this individual and having difficulty finding intact HIV, which could be a very encouraging finding and something to watch out for. The primary results are published in Nature. And then this trial you'll hear a lot more about on the next webinar. It's a small study of of a type of gene therapy approach where T cells are genetically modified to be better at targeting HIV. And they have it's called a duo CAR because there's a second modification, which is also gives them an inhibitor to block HIV from being able to enter the cells. So for the CD4 cells, that means they're protected from infection. The 9 participants in the study so far, it's sponsored by UCSF. It's happening at University of California, San Diego. That 9 people have undergone an ATI, and the media coverage that happened recently was really focused on 2 participants who'd started antiretroviral therapy quite early after HIV acquisition, who received the CAR T cells, and then when they underwent treatment interruption, they are still, is my understanding, controlling viral load to low or undetectable levels. In one case, for over 1.5 years, and the other for getting on for a year. And so there's some excitement there that modifying T cells to be better at targeting and killing HIV infected cells could be a beneficial approach. And the fact that people are now seeing, researchers are now seeing more cases of people controlling HIV viral load after a treatment interruption has allowed for looking for what they call correlates of what's going on, immune responses that may be contributing to this phenomenon. And there's been a couple of important published studies that have looked at multiple trials, including the small UCSF study, and many of the trials at the University of Aarhus. And there's a type of killer CD8 T cell response that if the CD8 T cells are able to respond very rapidly to viral rebound and proliferate, copy themselves extensively, so kind of generate a swarm of CD8 T cells all focused on HIV, that seems to be a very important factor in leading to control of viral load. And there's a word that the immunologists use called stemness, which seems to mean that the CD8 T cells have a kind of youthful and young profile. They're able to proliferate a lot more than older CD8 T cells that are getting a little exhausted. And that exhaustion is actually an immunological term. It's one of the few immunological terms that actually kind of means what it says, which I always appreciate. And so trying to get people's CD8 T cells to have this more youthful profile could be something to pursue. There's also evidence of enhanced antibody responses, like mentioned earlier, people's own autologous antibodies doing a better job of contributing to HIV suppression, reductions in reservoir size. And although it's kind of technical, there was a really great review published recently by Demi Sandel, Michael Peluso, and Rachel Rutishauser at UCSF, really looking through all these studies and reviewing what's been reported in terms of what's contributing to this good outcome in the people that are being able to control without ongoing treatment. And shifting again from those encouraging findings to interventions maybe where there's reason to hope that there might be encouraging findings in the future. This particular approach has a certain appeal because it involves a class of drugs that's already very well known, the anti-HIV drugs called non-nucleoside reverse transcriptase inhibitors, or NNRTIs. And it's this kind of strange finding where researchers discovered that some members of this drug class, the way that they inhibit HIV, it alters the viral life cycle in a way that usually when HIV is replicating, and it sort of integrates in a cell, uses that cell to generate more copies of HIV, and it uses something called the protease enzyme, which you've probably heard of from protease inhibitors, to chop up the new viral particles, create new virions to release from the cell. But the protease bit only happens when the viruses are exiting. That's kind of how the viral life cycle works. And what these drugs do is cause that protease enzyme to appear earlier when the virus is still inside the cell. And it turns out that there's a kind of innate antiviral mechanism that broadly recognizes proteases. And when that mechanism recognizes proteases in the cell, it kind of sets off a booby trap, which causes the cell to die. And so these drugs appear potentially able to specifically cause the death of any cell that contains HIV that's kind of trying to make more HIV that's kind of a little bit active. And we know that the HIV reservoir in people can, even though replication is stopped, it can show some activity and could be vulnerable to this kind of approach. And so Merck has published a paper with laboratory and presented some mouse results at CROI, and they've now advanced a candidate really designed to promote this kind of activity called MK4646. It's in a phase 1 study in Moldova and Romania right now. There was a study, I think last year or the year before, in Belgium in HIV negative participants. There's no results as yet, but I think this is something to kind of watch, because in theory in an RTI, it's a fairly standard part of an ART regimen. And so you could have a component of an ART regimen that's actually accelerating the depletion of the HIV reservoir, and a low HIV reservoir size is associated with a better response to all the other kind of BNAbs and other interventions, CAR T cells. So you could hopefully, use this as a method of reducing the reservoir and increasing the chances of someone achieving long-term suppression without ongoing antiretroviral therapy. There's a little bit of controversy around whether licensed existing NNRTIs can mediate this activity in people, whether they get to high enough levels in the body. But there are studies. There's an efavirenz study ongoing in Toronto and a study from Seattle that was presented at CROI this year, suggesting you might see some of this activity. And a study in China is now looking at riplivirine as well, and specifically looking at whether this activity could help reconstitute CD4 cells in people that don't get good immune recovery on a standard antiretroviral regimen. So I think we will be seeing some results from this approach fairly soon, hopefully. And then this was sort of mentioned in the promotion for this webinar. It's a kind of alternative strategy to trying to get the immune system to clear or control HIV. Maybe there might be ways of getting the virus that's integrated into a CD4 cell to just to be locked down and inert so it can't rebound. It's a little uncertain whether this is possible, but there's a variety of compounds that people are looking at that might be able to contribute to that type of activity. There's only one ongoing study right now, and it's of I'm not even sure if I could pronounce this properly, spironolactone in Brazil. The principal investigator is Esper Callas. And again, one of the common uses for spironolactone is actually in transgender people for feminizing purposes. And so again, this is something where transgender women taking spironolactone, studies could be helpful in people with HIV. But again, the possibility of that is going to be foreclosed if all this, institutional hatefulness isn't stopped. Another recent drug, and I think a recent webinar, or one of these webinar series covered this, the anti-diabetes drug metformin might be able to have this kind of block and lock activity. There's no ongoing trials right now, but there have been past studies in people with HIV that are being analyzed to look to see whether they might find evidence of this kind of locking in activity. And then, I think it's difficult to kind of cover all the potential challenges here. And I think there's an amazing group of people on this call. I think I'm hopeful can fill in the sort of blanks and things that I've managed to miss along the way here. Participant in geographic diversity is a key thing because we need an HIV cure that works for everybody. And so it's really important to study candidate interventions in a really broad population. BNABs have shown a lot of promise, but this requirement for screening and potential for resistance is problematic and suggests that there could be a lot of individual variation and need to broaden the efficacy of BNABs to cover more people, develop new BNABs or ones with alternative targets. The continued need for treatment interruptions isn't really ideal because it can be a nerve-wracking experience. There's a lot of education around the importance of staying, adherence and staying on antiretroviral therapy, and treatment interruptions can seem a bit contradictory to that. Long-term risks are still a little bit of concern, even though these treatment interruptions are much shorter than the older style of treatment interruptions that used to be studied. There is a proposed cohort of people who've participated in this type of study to monitor for any long-term health risks that I'm not sure if I don't think it's underway yet, but it's being proposed by UK researchers called COAST. And hopefully that might help kind of provide reassurance that there aren't any sort of long-term implications of interrupting treatment on a short-term basis. I haven't talked, there's a whole lot of really excellent social science related to HIV cure research, both in the scientific literature, and adequate support for that and integration in studies is really important. And then, getting to where we are politically now, unfortunately, we're seeing just an incredibly unprecedented attack on science. We've had grant terminations related to HIV for studies involving transgender people. This sort of people trying to, the political people trying to target DEI, even though they don't really understand what it is. And that's got some of the work trying to improve diversity in clinical trials has got caught up in this. It's irrational, it's bizarre and hateful, and I did sort of exactly how we get beyond that I don't know, but the midterms maybe could change things a little bit. And the US Office of Management and Budget is really, Russell Vought is a zealot who is trying to institute strict political control over federal grant funds. And we've already seen that this administration in the United States is willing to use federal grant money, or to try to use federal grant money as a tool for extortion, to get people that are doing things that they disagree with to stop doing those things by withholding money. It's evil and wrong, and it's really important that people try to do whatever we can to stop this. So far, that the effects have been on the kind of the people supporting clinical research, that there were effects last year, I think some of which have been addressed temporarily, but there was a withholding of funding from the networks for a while. There was some pauses in HIV cure-related protocols. I mentioned that Rio was the largest BNAB trial to date, but there's a larger 135-person trial with sites on the African continent that was paused, but I believe is now fully enrolled. Withholding of NIH funds to support research in South Africa because the administration is racist. And, broadly internationally, these new OMB regulations are trying to stop or really restrict international work, which is vital for science broadly, not just HIV research. Um, and that, I think has accounted for a little bit of regression in terms of the geographic reach of cure research at the moment, and why it's not expanding so much. And I'm going to make a plug here, and I should say this is coming from me and Treatment Action Group, and not any of the other sponsors of this webinar. Um, there's a public comments period open until July 13th on this proposal. There's some great resources from Stand Up for Science. AVAC issued an action alert. TAG will be issuing a statement shortly. IDSA and Infectious Disease Society of America and the HIV Medicine Association have created a good bullet point summary of the worst aspects of this, and also have a form that will automatically, hopefully, if it's working now, that you contact your congressional representatives, because OMB isn't necessarily going to, as it's currently configured, isn't necessarily going to listen to these comments, but Congress people can add to pressure, and perhaps, if it becomes possible, consider legislation to stop this kind of political takeover of federal funding from happening.
Mhm.
And then just to end with some acknowledgements, and maybe a caveat that there's just so much HIV research going on. I may have missed things. There may be important things that I haven't covered here. And just thanks to the organization that has employed me for a few decades now, Treatment Action Group, and support from funders, Foundation for AIDS Research, and I think just it's the Gates Foundation now. And I really appreciate all the researchers and trial sponsors that do actually register their studies, because it's not always required, but it's so helpful. We're trying to track what's happening with the field. And also just the people that are very diligent about reporting participant demographics when they present results. And I'll stop there so that we can discuss, and people can raise issues maybe that haven't been covered here.
Mhm. Siggy, you have a question.
Yes, thank you for this excellent presentation, Richard. I think there's very little to add. But actually, for the challenges, I'd like to add one thing that bothers me a lot, because for all the approaches that modifies T cells or that genetically try to modify stem cells, you need a kind of conditioning, which is just a nice word for chemotherapy. And it comes in like different intensities, from very unpleasant to almost deadly. And depending on what cells you're trying to modify, for somatic T cells, you don't need this intensive conditioning, but for stem cells, you need basically myeloablative conditioning, which means you kill the remaining stem cells in your body. And that leaves you for at least 2 to 3 weeks without any immune system, which of course poses you at some danger. And I think we have to look very careful at all these cure trials and proposed cure trials that are using conditioning in some form or other, because it really is some risk to the patients.
That's a great point, Ziggy. Richard, on those challenges as well, I mean, there's a couple of things that I continue to, yeah, I don't want to say bark, but I guess that's what it is. Just for instance, BNABs and the resistance issue, I mean, they're not going to be helpful for a great percentage of people. And we continue to see trials where, okay, everything works for a few people, and then they continue to use that compound. And I really don't see a lot of research on why they only work for some people. I mean, I think we really need to dig into what it is that we need to change. Is it the person? Is it the intervention? What is it that we need to do to make these more, worthwhile for everybody? And the ATIs, I mean, people are there. We've been doing small studies, and once we need to get to do big studies. Lots of the people who've already done an ATI study, I doubt we're going to see them again. And there are going to be lots of people who don't want to go off of their medication. I mean, if they understand what the study is about. So, I mean, and we don't really see, every once in a while, there's a new assay study, or we hear a little bit here or there about surrogate markers and things we can do to maybe not need ATIs in the future. I mean, I know that's a huge subject, scientific area, but we just need research in those areas. The other thing is, something that Jeff Taylor and I did. I mean, we've been doing these webinars for in collaboration with Dare and Rid for what? How many years now, that Jeff? A really long time. And the
Five,
first time, go ahead.
five,
Yeah.
5 years ever since the grant
Yeah.
started with Red.
Yeah. The first time we were in, we did a live one with people, at a center as opposed to a webinar. I mean, it really came out that, at first people were like, okay, what? No HIV? And then it started to dawn on them that, okay, no HIV, no housing, no eligibility for care, no eligibility for drugs. So, I mean, I know that this is far into the future. But we really need to think about, the policy concerns of what we're doing here. If we're successful, we need to think about what the landscape is going to look like for people. And on that OMB regulation thing, I mean, people need to look out. There are going to be lots of outreach to ask you to get in touch with your helpful outreach. Hopefully, that'll make it easy for you to make comments, and especially your Congress people, because as Richard has indicated, we've done the comments before, and I just think they're in a big trash can somewhere sometimes. But anyway, so please, if you get a notice with that sort of information, just distribute it widely. So Richard, I know I said a lot there, but and I know there's no answer for them, but mostly, I'm just talking about holes in things that we're not thinking about enough, I think.
Yeah, and I think, for some of the ATI concerns, excuse me, there is some progress, I think, in potentially identifying biomarkers that might help predict that someone's more likely to control after a treatment interruption. That thing about CD8 T cell youthfulness, it seemed to be enhanced by the interventions that included BNAbs. But if people had more of that at baseline, they were more likely to then control. So, sort of getting towards things that might help, either select who's appropriate for an ATI, or ideally get to a point where you've adjusted the biomarker and it's going to predict that someone would control. That would be the kind of holy grail, I think.
Yeah.
Michael has a question.
Michael?
Hi, and thank you, Richard, for doing this webinar and for all the work you've done on cure research for so long. Many of the studies whose protocols I've seen focus on people who are newly infected or who began treatment very soon after acquiring HIV. From looking across all the variety of studies, do you have a concern that those of us who have been living with HIV for decades might be excluded from the research as a whole, and or the cure product that eventually comes out of one or more of these trials? And how can we address that with the research establishment to be sure that we're not limiting the breadth of whatever positive outcome eventually arrives?
Interesting. Yeah, I think there was some focus, the part of the reason for that focus is this kind of idea that people with smaller reservoirs are would be a good starting point. But I think I just got asked to extract all of the acute infection studies for an event that's happening soonish, and it wasn't quite as big as I was expecting. I think it ended up being 12 pages instead of 50, so although there's focus there is still a lot of these studies are just recruiting people that have been on antiretroviral therapy for X number of years, and they don't have criteria for when you start it. So, there is information being generated, and but like, the Rio trial, which did seem to have that result, that was all people with primary infection. And I think that some the trials that have included people studied later have been a bit more like, there's been individual cases, but it's not been quite such a high proportion. I think it's really important, and I know Jeff and Linda and others have been advocating for this, that people don't forget or leave behind people with longer term infection, because it's a crucial population. And I think that there may even be certain interventions that could work better, because if you've accumulated a bit more T cell dysfunction, then an intervention that's designed to sort of correct T cell dysfunction may have a larger pool of T cells to sort of switch back on in someone that's had a longer term HIV, which is a bit kind of counterintuitive, but there might be some potential there. I would just agree that it's really critical that we don't just kind of leave behind people that have been having to take antiretrovirals for decades now.
Siggy.
Yes, in addition to that, just recently some research came out that shows that people who are on antiretroviral therapy for a very long time, which means more than 15 years, they have actually first signs of kind of rejuvenated immune system. They'd call this stemness. That was Richard mentioned in his slide set, and that may point in the direction that perhaps people who have been on antiretroviral therapy for a long time are also good candidates for at least some of the cure approach. Probably not for all of them, but the size of the reservoir obviously is only one factor. The strength or the stemness, the youth of your immune system is another one.
We need to remember too that if you think about it, I mean, people that have been doing this forever, this is exactly what we faced in antiviral research in the beginning. Oh, we can't throw out the baby with the bathwater. We've got to make sure that we're going to do this in people that they don't really say it, but that'll have better results. And, we don't want to hurt people either. That's a really big thing. So what we've been doing, Jeff and I and a number of other people going through protocols and stuff, trying to raise the eligibility criteria, the age eligibility criteria, and to go through the protocols to make sure that people are, in other words, we're not doing any harm in the ATI part of it for people who have been on therapy for forever, people with chronic HIV, and also to make it make that necessary and not really unnecessarily broad, so that people are included. The other thing is this block and lock stuff and the metformin. I mean, we had a presentation that with Hope that was amazing, that really talked about all this aging and immune system and that whole area. I mean, that we could go on forever. It's very complicated, but you could see where it would be important to do trials just in people in long-term survivors with those sorts of new things. So, I mean, we're working on it, but it's really very hard. A lot of it is just the protocols are copy and pasted. That thing from Mark Milano was always screamed about, that people just copy and paste protocols, and it just, so many people are not eligible just because, somebody wants to be quick about putting a protocol together. So it's multi, faceted as are many things, but we're on it. It's just difficult.
Yeah,
No, I
part of
think
it is people just make assumptions about what aging means, and somebody who's 60 or 75 can be just as healthy, if not healthier, than somebody who's 50. So again, you need to be scientific criteria and not just these blanket categories.
And there is some evidence, I think, that advocacy has been effective, because when I last looked, and I should have done it again for today, is that there's been definitely been an increase in the number of protocols that have no upper age limit anymore, and also what looked like a slight shift from sort of 60, 65 to 70 or above. So I think that has had some impact. And the other encouraging thing I mentioned, that there's these assays, these tests now that can help distinguish intact and viable virus from virus that's defective and probably not capable of replicating. And what you see in people that have been on long-term antiretroviral therapy for decades is that the intact virus is being preferentially depleted. So even though there's been no additional intervention, the residual immune responses and maybe those restored CD8 kind of responses that Siggy mentioned are actually kind of mowing the reservoir lawn while people are on antiretroviral therapy. And part of the encouraging thing about the TACK molecules is that maybe they could do that even better. And you could get really significant declines in the reservoir from what would be, a fairly standard antiretroviral drug combination. And I did see actually that Michael, you had some questions about TACK molecules in the chat.
Yeah, I did. Before I get to those, though, I actually wanted to. This is another question, and I know Mark Frank has been waiting to ask it.
Sure.
You had 11 people listed as being cured from the stem cell transplants. He has a list that's 12. I was wondering if you could pull that slide up again so he could look at it and see if there's a person that's been missed or if there's some other discrepancy going on. I just thought that would be. I would like to know that because I know that.
Sure thing.
Numbers.
If I got it right, it's the Essen patient, and I think he has not been published yet.
Okay, cool.
That's probably the reason why.
Sounds good to me.
He will be mentioned in Rio.
Great.
He's
So
spilled
we'll
the beans.
We've got a lot to thank Germany for, right?
It's the Essen patient. Yes.
Okay. Great. And so do we know, like, my one question about TACK was like, are there any like biological reasons why some individuals might benefit while others do not? Like we were just talking about this. And so like, I was just wondering like if we have any evidence like that, or is it still just all in dishes and we don't know answers like that yet because we have to get it into human beings?
Yeah, I think that's considered sort of unlikely because it's a thing that's present in everybody's cells. There's this thing called CARD8, which is some sort of sensor that senses the protease, and I'm pretty sure everybody has it. But I don't want to be too certain because I'm not a scientist.
Right.
But there was a presentation, I think, by Louis Montana at Croy from some laboratory work that there could be some limitations for how many cells that this approach could get at. But it was a bit technical. I'm not sure the details, because it would need virus to at least be sort of attempting to sort of generate, trying to make some RNA from its DNA or trying to make some protein. It needs to be doing a little bit of something, even though antiretroviral therapy is stopping it replicating. It needs to be trying to be active in order for the TACK thing to work.
And Rachel had a question for you. She wanted to know if there are any new HIV medicines that are being worked on for long-term survivors that may not qualify for these cure type studies. In other words, newer treatments.
There's definitely the progress, I think, towards 6-monthly injectable regimen, because right now we only have lenacapavir, the 6-monthly, and that's a Gilead drug. And, they would love to be able to partner it with something else to have a regimen you only had to take twice a year. They've been trying it with a couple of BNABs, but that brings up all those issues around BNAB resistance and having to screen people. And like half the people that tried to get in the study couldn't get in because they had resistance. So that's probably not ideal for a 6-monthly thing. But they just started a new trial of lenacapavir plus a drug that I think is codenamed GS3242. Which they hope lenacapavir is a capsid inhibitor, 3242 is an integrase inhibitor, which they think they can get to every 6 months. So that could be like the first twice yearly regimen, which, people vary and, injectable might not be a preference for everyone. But if people are okay with that, it's going to the clinic twice a year instead of taking daily pills.
Mhm.
would be
You know, Richard, you
progress.
just gave a fabulous real world example of the problem with BNABs. You know, how are we going to use them? Who are we going to use them for? What we can't put them in this trial because, you know, I mean, all this work, all this money, they may be pretty useless.
But it's not, I mean, I think, my only counter to that would be just that it's not a static kind of field in that people are still trying to discover sort of new and better
Yeah, I agree with that.
apps. I mean, that might not still cover it, but, the ones that are being used at the moment were discovered quite a few years ago. And they're, the BioNTech one I think comes from Florian Klein's lab in Germany. And it's particularly potent. So there may be room for improvement there.
Yeah.
Mhm.
Yeah, I hope so. I just think that sometimes we get stuck, you know. I mean, we're not in a, well, look at your last slides about the money and how it's being, you know, ugh, I don't even want to go there. But, you know, we need to really think about, okay, wait a minute. We can continue to study things, but we need to look at other things as well, is I think my main point, and stop, you know, we're not ACT, DDI, DDC, we can't be in that 40 years from now stage is what I'm saying, my point.
Mhm. Did anyone else have any other questions they wanted to bring up, or a topic or thoughts that you were having about anything?
Mike, I
Go
just
ahead.
wanted to comment on the BNAP thing, because I think they are kind of underestimated right now, because most people look at BNAPs as a kind of antiretroviral drugs on steroids or something like that, mainly because they're so expensive. But we mustn't forget they are molecules of our immune system, and they don't only bind to the virus, but they only bind to cells of the immune system, and they can change something in the immune system. And they may be part of the success story of the BNABs that may lead to prolonged suppression of the virus, even when the BNABs are already out of the body, you know? So I think there's still a lot to learn. And even though the BNABs we have today are not perfect, we probably can overcome that with selecting even better BNABs or combining even 3 or 4 BNABs. So far, we have been trying only 2. And there are also ideas that we could combine the binding sites of 2 BNABs into 1, or even 3 into 1. So there's still a lot of going on in this BNAB field.
Paying for all those BNABs.
What?
Who's going to pay for all those BNABs?
Well, right now they are pretty expensive, but the thing is, if you have a BNAB that you have to give, like, let's say every 6 months, because we know how to biochemically adapt them so they can stay in the body for 6 months. And after that 6 months, you have a kind of BNAB-induced immune control for another 12 months. Then you have to give it only every 18 months. And even though the BNAB itself is kind of expensive, you have to give it only once every 18 months. So it could be cost effective in the long run.
I mean, I've been waiting for that promise of that vaccinal effect for a really long time, and I just don't see it.
I mean, there was evidence of enhanced T cell immunity in the Rio trial participants associated with BNABs that wasn't seen in the people that didn't get them. So that is some. I mean,
how long does that last?
You can maybe not confirmation, but I mean, it's contributing to the people that have been off therapy for 72 weeks.
How long does that last? I mean, I think that's the promise was that the promise was you were going to get 1 dose, and then it was going to keep continuing in your own body, and you'd never need it again. And now then we need 2, 3, whatever, and then how long does it really last once it's in your body? It's just and I'm not saying to give it up. I'm really not. I'm just saying that they really need to hone down on what's going on here instead of just throwing them in, you know, with all the bathwater for every trial without doing more work.
Mhm.
We need to learn about the bridges while we are crossing them. That's the problem here.
Yeah.
I agree with you too.
Cancer dealt with this. I just wanted to point that out before I go on to Mark's question. Is like cancer, you know, first it was like there was going to be a cure, but now we know that we have to do a lot of work to sculpt the thing that will work as the best treatment for some people, and some people are left out. But it took a long time for cancer to learn that. And I think that HIV researchers, I don't even know if we have any here, but they need to start building in those questions. Like, it's not only important to know what happens to the people who respond to whatever therapy it is, but what about the people who didn't respond, and what are we doing for them, or what other sorts of things that we're trying? And I think if we just even heard that from researchers more often, I think we'd feel better that they're not forgetting other people and
That's
leaving them
what I'm
behind.
talking about.
Yeah, I just think that too. So go ahead, Mark.
With the NRTIs, do you know if, I'm assuming you wanted me, right?
Yeah, no, Mark, I didn't see that you're there, but yes, Mark, you go on first, and then we'll get the other Mark. Two Marks, both spelled with a
Everybody's
C.
named Mark.
I'm sorry.
No, please, it's not your
stop.
fault. I was just looking at Mark Franca, and that's why I was like,
I
wait.
am so sorry. Do you want me to
Please.
hold off?
No, I want you to
Go
go.
ahead.
Okay. With the NRTIs, do you know if any of those that were having their effect more broadly had purines in their structure, meaning like adenosine kind of molecular structure? Because some of the HIV medications, and I've brought this up even at NIH before, is that, these medications could ultimately have other effects just because of their chemical structures and their ability to bind to cells in other ways that affect the signaling pathways. So, I wonder sometimes that we may be shooting ourselves in the foot and actually aiding persistence by, some of these medications doing off-target effects.
And
I
I
Can I
talked to the
answer
Merck
that?
people and tried to, and try to get them to, they're not ready to talk to us yet. But, if that's actually a really good point, Mark, because if I could mention these other sorts of preclinical type, well, not preclinical, but, different sorts of questions that I'm sure they must be looking at now, before they get in and, out of phase one. I think maybe I could convince them to come and talk to us.
Sorry to jump in here, but I think Mark was relating this to the TACK molecule, right? And TACK is derived from the NNRTIs, not the NRTIs, which are nucleoside analogs. And some of the nucleoside analogs had purine bodies in them. So the question you basically ask is about nucleoside analogs. And as we have seen in the past, they have a lot of side effects because they bind to more than just HIV or reverse transcriptase in the body. And that's what causes those side effects. We have learned that the hard way.
Mm-hmm.
Right.
But the NNRTIs don't have purine bodies in them. They have a completely different chemical structure.
Okay, because that's important for us to know.
Yeah.
Yeah.
They have other
Hey,
problems,
thanks,
though.
Siggy. I would know. I wouldn't have been able to answer that.
Mr.
So Mark
Franca.
Frankie.
Frankie.
Now the other Mark.
Yeah.
I only wanted to mention that I met Professor Florian Klein and his team in Cologne last week, and I met the guy who is on ATI for 8 years now. That is the longest person living after an ATI after receiving BNAP. So I think this is very promising. And I think the Klein Lab is also involved in the Rio trial.
Okay.
I saw Rio on several slides last week.
I think because I think they want to do that, they're going to also add an arm that looks at people doing more than one ATI in the Rio trial. And so I think they've added sites in a number of other European countries, including the site in Cologne.
Maybe we should ask people on the call what they think of ATIs. Would they be willing to get in a trial with an ATI?
Well, I had one, and a
You're still
long
here,
one.
right?
And
That
the
worked out
problem
pretty
was
well.
that
Worked for you.
there was only Timothy's case to compare. So my doctors cared a lot about me. So I wasn't the one to stop art as the second after Timothy. And you have to rely on your doctors. And I also heard from the patient from Cologne that he had the art at home. So every time when he went to the HIV test to the university hospital, he was prepared to have a call and to start immediately with art. So, yeah.
I think
And
that's
that
really.
was Björn told to me, if you have it again, you start art and everything will be fine. And I relied on that.
I think it might be helpful if they could develop an at-home sort of
Yeah.
test that would let you know if viral load is rebounding.
Yeah, we've been advocating for that for a while now. It's not ready for prime time.
Yeah. Mark, let me ask you what the protocols are like in Germany. How long, there's always this, tension here about we want to, initially we tested people twice a week, once a week. And then there's the other side of that, dragging people in every 5 minutes and people not wanting to come in, to the clinic. So what do they do in Germany? What's the usual protocol? Once a week, every other week?
There was no protocol because I was the second after Timothy. I know that there are concerns from the ethics team to stop the art. And first, we started twice a week, I think, for 2 or 3 months, then once a week, then every second week, then every month, then every 2 months. Now we are at 6 months. And yeah.
Well, Ziggy, do you know the answer
Linda,
to that?
yes, I can tell you about the planned protocols, and they have planned a viral load test every week in the first 16 weeks, I think. And to make it easier for the patients, they are offered study visits by nurses that come to them, to their home, make the blood draw, so they don't have to come to the clinic. Because we have only so many study centers in Germany, and you cannot tell people who probably just had a stem cell transplant to travel like 800 kilometers on a heavily visited train, with a reduced immune system. You just can't do that.
That's a great idea.
But you have to really put a lot of effort into that and to really think about it to make it easier on the patient and still be manageable,
Yeah.
Because daily blood draws are not doable.
Yeah, they're just not.
I think for a 135-person study, it could get quite complicated.
Yeah, but we're talking about 8 or 9 patient studies right now. And still, it's quite challenging and expensive, of course. And the other thing I put already in the chat is that the researchers are very reluctant to also include psychosocial research and care for the patients, because I think especially during the ATI phase, that's very important for the patient, that they feel really seen and that they have the impression that their problems and their thoughts are really taken into account. And researchers just don't see that. They see, well, if the viral load goes up, we restart therapy. Yeah, but that's not all of it, That's not the dimension for the patient. And we really, as patient advocates, we really have to stress that and really have to push for that.
We've done a lot of work in that arena in the United States, and I think we've really come far. I think Karine, I was actually just looking to see.
You have Karine, we don't have anybody.
Yeah, I was just looking to see if she was still on the call before this came up, but I don't see her. But we really beat the doctors up about, you have to have, all the way through the study, psychosocial sort of care or opportunity
support,
people, check in with them after the study to see what the story is. I mean, just imagine being off your therapy, being on the edge of your seat for the whole time. I mean, it's
and one thing we haven't even talked about is, if there ever is a cure, will it prevent reinfection? Because as we've seen in hepatitis C, about 30% of the people being cured get reinfected within 1 year. Probably not with HIV, because HIV is not as infectious as hepatitis C, but still a noticeable amount of people will get reinfection if there's no protection. So
Yeah,
that's an actually interesting question. As you probably all know, Timothy Brown took PrEP after he's been
Yeah.
cured because he was afraid of X4 trope virus, you know? So that's another important question.
Yeah,
Mark?
that's something that we don't. Yeah, that's very, very important.
Then Mark had another question.
Well, they told me that a student will join me twice a week because I had around 100 kilometers to the university hospital. And I said, oh, okay. But then they checked the costs, and I had to go to my local doctor and send the blood to Cologne. And later I learned how to pick the blood samples myself. So that was the easiest way to get the blood to Cologne.
So did you do a
Those-
finger stick on a piece of paper, or how did you take the
Did you
take the
draw blood or what?
Oh, they need vein blood.
Yeah.
Yeah, it's easy if you're practicing this.
Junkies do it every
I
day,
can
right?
testify it's not that hard.
It's not junkies. It's people who use drugs,
I'm sorry.
please.
Of course, yes. I'm sorry. Yes. Politically correct.
I didn't say that. Yeah, that's all interesting. We did a lot of work on a study that Merck was doing with another company on a home care thing, and we went through
So
all these issues, you know, but it just didn't pan out. I don't think it was effective enough, to tell you the honest truth, and we just haven't heard back. But I mean, it's something that we talk about all the time. I mean, the doctors realize it, but it's just, again, another spot that needs, guys, they can make a fortune for people with HIV, people with, it's just so necessary.
I don't think they could get the sensitivity down below like 1,000 copies. So it was
Maybe that was what the problem was.
yeah,
when you
It was
really
one of the
want it,
issues.
you want it to be down around 50 at least.
Yeah. They really wouldn't talk to us about it afterwards. We just knew it was a flop. We didn't know why.
When it was really onerous, you had to take the sample, FedEx it somewhere. Wait, it wasn't immediate, right?
Yeah, you
Yeah.
had to send it.
No, it was definitely a
It was
first
a
stage.
pressing thing that you put on your upper arm.
Yeah.
Yeah.
And sometimes it bled and it carried on, and then you couldn't get enough blood, and it was not optimal. You'd think somebody would jump on that opportunity.
I'm thinking the
I think I
applications
did a
of,
lot of research during COVID, but so far to no avail.
Yeah.
they could use that in a democratic DRC about now to fight Ebola,
Yeah.
right? So
Exactly.
I see the time running down a bit, so I just wanted to make sure to say thank you to everybody for all this.
Thank you.
Thank you.
Thank you.
This
Put a
was
lot of work
marvelous.
into that.
It was marvelous. It was such an excellent job. I mean, it was everything I hoped for, that's for sure. So while we just have 3 minutes left, let me just remind everybody again, July, what is it? 14th, right? Let me make sure, because one was 16 and one was 14. This is 16 and July is the 14th. Steve is a great speaker, and I'm sure we have a lot of questions for him for this CAR T cells and gene therapy idea. So
because I think there's aspects of that study that I didn't really get much detail on. Like they've done some gut biopsies on the two people that have got extended post-treatment control, and it sounded like they're having a hard time finding HIV DNA in their gut biopsies, which, if that's accurate,
that's
that would be quite unusual
that would be wow.
Tim Henrich has been doing the gut biopsies, and he's got a lot of experience with that. So
wow, I didn't know that. That's news.
But don't forget to ask about conditioning.
Yeah,
because that may be part of the story.
The one serious adverse event that happened was somebody that already maybe had slightly
cyclophosphamide, right?
Yeah. Might have been, I can't remember which it was, maybe neutrophils or something, that something went down too much, and it's recovering now, but it was a not great response to the conditioning. I don't think they've seen any problems with the T cells. It's just the conditioning.
Well, Vicky, you're going to have to get on the call next month too. That's
I'll try. I don't know if I can make it, but I'll definitely try.
yeah. Well, we'll carry that your message for sure. So, anybody else? 1 minute to go. Anybody else? Questions, ideas, thoughts? Well, I guess we're right on time. I love it. Thanks again,
Everyone.
Richard. This was just marvelous.
Sure thing. Look forward to the next one.
Okay. All right. Goodbye, everybody. Take good care.
Bye,
And look out for
everybody.
that OMB stuff, okay?
Yeah.
We're gonna make it so you can just sign, click this and all that sort of stuff.
You got it.
All right. Thank
All
you.
right.
Bye.
Bye-bye.
Ciao.